Welcome to Professor SNAB, where we aim to make A-level biology as simple as a cell. So today we're looking at understanding the process of DNA replication, including the role of DNA polymerase. We're also looking at how Michelson and Stahl's classic experiment provided new data that supported the accepted theory of replication of DNA and refuted competing theories. So let's look at DNA replication. So copies of DNA are needed when a parent cell divides. Copying DNA ensures that both the daughter cell will receive full copies of the parental DNA. The DNA is copied through a process called semi-conservative replication. It is named as such because in each new DNA molecule, one of the DNA strands is from the original DNA molecule, while the other strand is newly created by the cell. The new DNA molecule has conserved half of the original DNA and then used this to create a new strand. This ensures genetic continuity between generations of cells. This also ensures that the new cells can continue the role of the old cells being replaced, and this also takes place during growth. So now we're looking at semi-conservative replication in more detail. So DNA replication, as we know, occurs in the S phase, which is the interphase of the cell cycle. The enzyme helicase unwinds the DNA double helix by breaking the hydrogen bonds between the base pairs of the two antiparallel polynucleotide DNA strands to form two single polynucleotide DNA strands. Each of these single polynucleotide DNA strands acts as template to form the new strands made from free nucleotides that correspond to the exposed DNA bases by base pairing. The new nucleotides are then joined together by the enzyme DNA polymerase. The original strand and the new strand are joined together through hydrogen bonding, resulting in a new DNA molecule. This form of replication is known as semi-conservative replication. Looking at DNA polymerase, so there are free nucleotides in the nucleus, and these nucleotides contain 3 phosphate groups and are known as nucleoside triphosphates. And just to let you guys know, nucleoside triphosphates and nucleotide triphosphates are exactly the same thing, so you can use either names. The extra phosphates activate the nucleotides, enabling them to take part in DNA replication. The free nucleotides bind to their complementary bases on the template DNA strands. DNA polymerase joins these two strands through a condensation reaction between the deoxyribose sugar and phosphate groups of the adjacent nucleotides between the new strands, resulting in the sugar-phosphate backbone of the new DNA. The energy released from the two extra phosphates is used to create the phosphodiester bonds between adjacent nucleotides. Hydrogen bonds then form between the complementary base pairs of the original strand and the new strand. Leading and lagging strands. So DNA polymerase can only build the new strand in one direction, which is from 5' to 3' direction. DNA is unzipped the other way around, which is 3' to 5'. Hence, DNA polymerase will attach to the 3' end of the original strand and move towards the replication fork. The replication fork is the point where the DNA molecule splits into two template strands. The DNA polymerase enzyme synthesizes the leading strand continuously. The template strand is the same as the leading strand. The new strand is called the lagging strand. On this strand, DNA polymerase moves away from the replication fork, which is 5' to 3', resulting in synthesizing happening in short segments called Okazaki fragments. DNA ligase joins these lagging strand segments together to form a continuous complementary strands by catalyzing the formation of phosphodiester bonds and creating a sugar-phosphate backbone. So now we're looking at Michelson and Stahl's experiments. So Matthew Michelson and Franklin Stahl came up with the theory of semi-conservative replication. Bacteria were grown in a broth that contained heavy nitrogen isotope. As the bacteria replicates, nitrogen from the broth is used to make new DNA nucleotides. It was observed that the culture of bacteria contained only heavy nitrogen in their DNA. A sample of this DNA was extracted and spun in a centrifuge. The DNA containing the heavy nitrogen settled near the bottom of the centrifuge tube. The bacteria only containing heavy nitrogen was extracted and added to a broth containing light nitrogen associated with 14. After one round of DNA replication to take place, their DNA was extracted and spun in a centrifuge again. The DNA was found to contain both heavy and light nitrogen, hence finding the DNA in the middle of the tube, hence proving that semi-conservative theory as the DNA strand contains the template strand and the new strand. So the template strand is from the nitrogen isotope 15 and the new strands from the light nitrogen, which is isotope 14. So that is all from Professor Snap today. Hope you found this video useful. If you have any questions, please let me know in the comments below and I will get back to them as soon as possible. And don't forget to like, share and subscribe.